There was a hard-working farmer who owned a farm that was home to a group of rabbits. These rabbits in particular were black in color. He knew that half of them were homozygous black, while the other half were heterozygous black. The farmer wanted to breed only white rabbits, so he decided to buy a homozygous white rabbit to mate with the black rabbits. He planned to sell any offspring that were not white. In the first year, the farmer bred the homozygous black rabbits with the homozygous white rabbit. Eventually, he sold all 80 black rabbits that he had bred. Next, he bred the heterozygous black rabbits with the homozygous white rabbit. This time, he sold 21 black rabbits and 19 brown ones. In the following year, the farmer decided to breed only the original white rabbit with the white rabbits from the previous year, and did not sell any rabbits that year. Now, the question is, what would be the genotypic ratio of the white rabbit's offspring?
Let's discuss. First, let's jot down the information we know. Farmer has a black rabbit from which half is homozygous and half is heterozygous. Second, the farmer bought a homozygous white rabbit. Know that there are two different genes that are independent of each other. We will set black homozygous as big B, big B, big W, big W. Heterozygous black as big B, small b, big W, small w. And homozygous white as small b, small w, small w. Remember that the farmer first bred the homozygous white rabbit with the homozygous black rabbits. When we do a Punnett square, observe that crossing the two will give us all black heterozygous offspring. That is big B, big W, small w. This led to the farmer selling all the 80 black rabbits in the market. The 80 heterozygous black rabbits were then bred with the homozygous white rabbit. Let us find out what the genotypic ratio is by making a second Punnett square. Crossing heterozygous black rabbit and homozygous white rabbit gives us a genotypic ratio of 1 big B, big W, small w, to 1 big B, small w, small w, to 1 small b, big W, small w, lastly to 1 small b, small w, small w. Recall that the farmer sold 21 black rabbits and 19 brown rabbits this time. What about the number of white rabbits that he did not sell? We know that half the rabbits produced 80 offspring, so we should know that the other half needs to add up to 80. If there were 21 black rabbits and 19 brown ones, there should be 40 white rabbits that the farmer kept. This leads to a 1:1:2 phenotypic ratio. After analyzing the Punnett square shown in figure 1, one can conclude that it must be recessive epistasis. This is because it is the only way to obtain a phenotypic ratio of 1 black to 1 brown to 2 white.
What is recessive epistasis? It is when recessive alleles at one locus mask the expression of both dominant and recessive alleles at another locus.
Now, let's jump to crossing the homozygous rabbit with the white rabbits he kept from the previous year. Remember that he kept the 40 white rabbit offsprings, half of which are homozygous white and half of which are big B, small w, small w. He breeds the homozygous white with half of the white offsprings big B, small w, and small b, small w. We're going to make two Punnett squares for both the crosses. Small b, small w crossed with small b, small w is going to produce all 20 small b, small w offsprings. Big B, small w crossed with small b, small w will produce a genotypic ratio of 1 small b small w to 1 big B small w, and thus 10 of the rabbits will be small b w, and the other 10 will be big B small w. With this, we get 10 plus 20 small b w, and 10 big B small w, which gives us 30 to 10 genotypic ratio. To answer the question, the genotypic ratio is 3 small b w to 1 big B small w.
The European green woodpecker, scientifically known as Picus viridis, is a notable member of the woodpecker family due to its distinctive appearance. This avian species is characterized by its size, as it's considered a large woodpecker along with its striking green plumage. Some members of the species have the chest pattern such as round chest, heart-shaped chest, or no chest pattern. One day, a curious biologist wondered, how would we be able to tell what kind of gene interaction is happening, and what determines what chest patterns will be expressed, and if we can come up with a biochemical pathway from this information. First, we would have two parents with the homozygous traits mate, one with the trait of no chest patterns, another with a round chest pattern. This would produce an offspring with a heterozygote traits from both parents, with the phenotype of no chest patterns being expressed. From that interaction, we take the heterozygous offspring and have them mate with a similar heterozygous bird. Once this is done, this forms the F2 generation. And we see that it forms 36 with no chest pattern, 3 offspring with round chest pattern, and 9 with heart-shaped chest patterns. Now here is a cross table showing the different alleles shown for the two heterozygous pairs, with big C having no chest pattern, small c, small s, round chest pattern, small c, big S, having heart chest patterns. And this all forms a 12:3:1 ratio. And from all this information that we receive, we can tell that it has 2 gene pairs. From this, we can work out the biochemical pathway, which would first start off with no chest pattern combined with enzyme 1 and needing 2 recessive small c alleles to continue through the pathway and make the round chest pattern. And going further, we would need enzyme 2 with 1 dominant big S allele in order to get the heart pattern. We can see from the above pathway that big C, blocks the expression of big S, small s, which means that big C comes before S in the pathway. With all this information, we can tell that it is dominant epistasis based off of the gene interaction and the ratio being 12:3:1, showing similarities with the amount of offspring produced in the F2 generation.